Good evening, everyone. You are so brave to come out on the first really, really frigid night. It's delightful to see you all here with all your coats. Stay bundled. Happy to have you at the final installment of Crossroads of Ideas for calendar year 2019. My name is Laura Heisler, and I am Director of Programming for the Wisconsin Alumni Research Foundation, Director of Outreach for the Morgridge Institute for Research, and Director of the Illuminating Discovery Hub for the Wisconsin Institute for Discovery. So those are the three organizations that collaborate to bring you this series. They're all housed here in this building. And we created this series several years ago as a way of really living up to the promise of the space that you're in, which what we call the town center, to become a crossroads of ideas on the campus, connecting researchers from different corners of the university and engaging with the community on subjects that are important to all of us, both as subjects of research and things that really matter in our lives. And so the Crossroads of Ideas series is really one that tries to bring us together to have conversations around things that matter to all of us that just so happen to be the subject of groundbreaking research here at UW-Madison. So that's the nature of this series. Before I talk about how we're going to run this evening session, I do want to make you aware of the dates for the spring semester. I'm very excited that we have all the dates set. So if you want to mark your calendars, they're all on Tuesday evenings at 7 p.m. right here in this room. I can give you a sneak peek into the various topics. We'll be exploring the affordable housing situation in Madison and trends and how trends how the trends are going to evolve in partnership with the University Year, which is a program that connects with cities to help cities solve problems with using researchers at UW-Madison as experts. And so this will be in conjunction with Dane County and the city of Madison, and that'll be in February. In March, we're going to bring in, if you saw in the paper a couple weeks ago, a new partnership between the Nelson Institute for Environmental Studies here on campus and the City of Madison to prepare for climate change, especially around issues relating to water and flooding, and we'll be having that session in March. In April, one of the researchers at Wisconsin Institute for Discovery here upstairs in this building, who does research on psychology of color and has built a virtual brain, will be talking on April 14th. And then on May 5th, we're going to round things out with a look back at the legacy of Howard Temin, a Nobel laureate from UW-Madison, who was an iconoclast and a really revolutionary thinker, and that will bring us to the conclusion of this coming year. So if you got an email for tonight's event, you'll get an email for those. If you didn't and you'd like to, we can catch your contact information on the sign-up sheet just outside the door to my left. So let us know if you want to be in touch about these events and these dates. In terms of how things are going to go this evening, if you've been to one of these before, how many people have been to a Crossroads of Ideas before? Wow, great. Okay, so you all know the rules of the road. We capture these, we record these, we'll make the recordings available on the discovery.wis.edu page in a few days. Because we record it, we ask that you wait for one of our mic runners to reach you when we get to the Q&A. We're planning on doing that at about 7:15. We will end promptly at 8, and I understand that tonight's speakers are willing to hang out and talk to you for a little bit longer if you didn't get a chance to ask your question. So tonight's session is going to be a little different from what you might have seen if you've been to these in the past. Instead of having a speaker stand right where I am and talk through a lot of slides, our speakers have opted to do more of a fireside chat moderated by the gentleman I'm going to introduce right now, Professor Emeritus Jim Dahlberg, who is a longtime researcher at UW-Madison and also a member of the Morgridge Institute for Research Board of Trustees. So Jim will be setting the stage for this evening's discussion and moderating it and fielding your questions as we get to that portion of the evening. So with that, I'm going to turn things over to Jim Dahlberg.
So good evening. So this is going to be a very interesting evening with the cross-fertilization of different ideas in the clinical sciences as well as basic science, and I'm really looking forward to it. The people who are going to be speaking, Melissa Skala and Dustin Deming. Melissa was trained, got a degree, a master's degree here in biomedical engineering. I almost said my department. And then went to Duke, where she had a PhD and postdoctoral training at Duke, and went on to Vanderbilt, where she was professor at Vanderbilt for several years. And we were fortunate enough to recruit her to the Morgridge Institute in 2016, and she's a professor also in biomedical engineering and in the Department of Medicine as well, or not? Yeah, and the Carbone Cancer Center. Dustin Deming has been here at Wisconsin for throughout his training. He got his MD here and residency and then a fellowship here, and he has extensive training in oncology and specializing in GI oncology at that. So, and he's also, I could read a list of honors that both of them have, but you take my word that they both have a very long pedigree of many great honors. So we'll start off with maybe Melissa give a brief talk about what's going on in her lab, and Dusty can talk about some of the things that he's doing, and then we'll get down to the integrative part of the discussion. So do you want to come up here and we'll just... What?
You want to sit?
I'm going to sit.
Let's all sit.
We're all going to sit. We're just going to get nice and comfy.
You can have the middle.
Oh, he has to turn his mic on. I'll just start talking. So I'm Melissa Skala, and as Jim mentioned, I came here. I actually grew up in Washington State, so my childhood dream was to be an astronaut. And then I decided I didn't want to go through all that training. And what I actually loved was the optics. So the physical sciences, physics. I took as much physics as I could in high school. And I loved rainbows and sunshine, so photonics, you know, how light interacts with matter, how it interacts with the things around us and what it can tell us about the world became fascinating to me. So as I was an undergrad, I started working a lot in laser physics. And I actually happened to come to the University of Wisconsin for a summer research program because my brother was here in electrical engineering, and I love to sail, so he loves to sail, so we're very competitive. So we decided we needed to spend the summer racing each other, so I had to come here and race him on Lake Mendota. But I was working in a biophotonics lab here, so in biomedical engineering, that just means we throw a bunch of light or photons at tissue or cells, and we try to use that to understand what's happening in the cells. And this was a perfect interaction of the physical sciences that I loved and the desire to do something bigger than myself, which is to understand, you know, life and how it interacts and how it grows and changes. So my PhD was actually started here at the University of Wisconsin, but my advisor moved to Duke, so that's why I have a PhD from Duke. And throughout my studies, I've been looking at basically how light interacts with cells and how it can change the color of the light we get back, how it can change the properties of the light we get back, and it really tells us about the metabolism of the cells, so how the cells make energy. And it turns out that's very specific for cancer. So cancer has aberrant metabolic activity. That just means it creates building blocks for new cells, and it creates energy to create new cells in ways that are different from normal cells. So that just means that we can use this information on light-cell interaction to find out if a cell is malignant or not. And then later on in my career, actually after I moved here to the University of Wisconsin, I interviewed and I met with Dusty, and we have similar ideas about what's needed in cancer care, which is that we have to understand what's happening on a single cell level. So we have to know what single cells are doing in someone's tumor. And we have to understand diversity between patients, so how different patients react differently to treatment. And that's really the essence of personalized medicine. So it was natural for us to work together, and what we're doing now is combining the optical technologies that I develop in my lab with the cool single cell, single patient cultures that Dusty develops in his lab to try to get a better understanding of this unique diversity between patients and cells in cancer, right? That's sort of what we do, in a nutshell. But we'll talk more about it.
Dusty. So I'm Dustin Deming. I'm one of as Jim said, I'm one of the GI medical oncologists at the Carbone Cancer Center. So I did my initial training here and stayed on. When I first started in medical school, I thought I wanted to be a pathologist, and then I started a research project with one of our medical oncologists, and actually the research project got caught up in regulatory approvals, such that I never got to actually work on the research. But what happened was that freed me up to be in clinic with the oncologist, and so I got to spend a lot of time down in clinic, see patients, see the hope that the research I was about to do, how the research that I was about to do was providing hope for patients, and in addition, see how when I got into the lab, that actually helped fuel the day-to-day pipetting liquids, moving this from there in different test tubes. That was fueled by the fact that this was actually meaningful for a patient in clinic. When I completed my oncology fellowship, I myself was diagnosed with rectal cancer. So as many of you may know, there's an increasing rise of colorectal cancer, especially in young adults, and I'm one of those young adults. Now that was 7 years ago, and I'm now cancer-free, so I'm very excited about that, obviously. That has fueled a lot of what I now do in my career, meaning what I'm focused on is developing new treatment strategies for patients with cancer, and so this is colon, rectal cancer, pancreas cancer, but more cancer in general. And so what we're trying to do is actually understand how each patient's cancer is different. So for the last 50 years, the way we've been treating cancer patients is we take a group of patients who have a similar disease, colon cancer, pancreas cancer, lung cancer, breast cancer, etc. We figure out how far the cancer has spread. Is it a localized problem or is it spread through the patient's body? And then we treat all the patients roughly the same way with the same disease and same stage. But what we now realize is that cancer, for example, breast cancer, is not just one disease. And we know that the individual kind of genetics, or what makes the cancer cells, the aberrations or abnormalities in their DNA, can actually tell us a lot about how those cancers are going to act and how those cancers will respond to different therapies. So I'm part of a group now that we call our Precision Medicine Molecular Tumor Board, where we are actually getting patient samples and looking at the genetics of each patient's cancer. So we've now, working at the Carbone Cancer Center, signed an agreement with a company called Strata Oncology that actually provides this testing for free for all of our patients who have advanced cancers. So if a patient presents with any sort of solid cancer, meaning lung cancer, breast cancer, colon cancer, pancreas cancer, etc., we are able to get a sample of their tissue and send it to this company, and the company will send us back data telling us what's wrong with that patient's cancer. So what makes that cancer a cancer? And we're now trying to use that to develop new treatment strategies for specific subtypes of cancers. Now that has taught us a lot, and we're able to more personalize our treatments for many patients. But what we have the trouble is now that we're finding more and more cancers are different from each other, it's harder to do the clinical trials that we need to do because each subtype of cancer becomes rarer and rarer the more we divide it up. And so what we're trying to do is build better models in the laboratory so that way when we find something that we think is really exciting for patients, we can actually bring that to the clinic faster and for the right patient populations. And so Melissa and I are collaborating, growing patient samples. So years ago, patients, we would get samples and we'd put them on a plastic dish and hope that the cancers would grow on that plastic dish. Unfortunately, it's only about 5 to 10% of all cancers that would actually grow hooked onto a plastic dish. Now there's a new method that we call organoid culture, and we're going to get into more of this later. The general idea is that we can now take cancer cells and grow them in a matrix or what actually looks like Jell-O. So you can kind of think of, so there's very few families that do this, but my family still makes Jell-O molds with the fruit in the Jell-O. It's really the best way to describe what we're doing. So instead of making the cells have to hook onto the plastic, we can lock them like pineapple in a Jell-O mold. And that Jell-O that we have, we put lots of nutrients into it, so it actually helps the cancers grow. So with this technology, we're now able to grow somewhere around 80% of patients' cancers, as opposed to 5 to 10%. And so this is opening up a lot of opportunity for us to do research. But one of the hard things that we have with our research and why our collaboration is so important is that it's very hard for me to figure out, just looking at our cultures and using kind of standard techniques, how well our drug treatments are doing on those cells in the dish. And that's where Melissa's technology has really helped us a lot.
So can you tell me what fraction of your time do you spend actually talking to patients, and what fraction?
So I see patients 1 day a week, and then I do the other part of the week. Half of that is clinical trials, and half of that is basic science research.
So I guess you started talking about how we assess the cultures, which is sort of where I walk in the door and I say, "What you doing, Dusty?" So what he was saying is basically you can picture these little pineapples in a Jell-O mold, and what we're seeing when the tumor is not responding to the treatment is we see those organoids, they're called, they grow. And if they're responding to treatment, they shrink or they die, and you can actually see the cell death, right? But it's not always quite so simple. So what was lacking, so the great technology that people have developed for growing organoids from patient samples has created this new opportunity for technologies to be able to assess those cells and ask, how are they changing? Are they responding to treatment? Yes or no? Why or why not? And what can we do alternatively to get them to respond? So I think that's why we sort of have a dual edge problem here. We have too many samples, we have a need for automated assessment, and we have a need for quantitative assessment so we can do more drugs and more samples basically at once. And it might be, actually, do you want to talk about how the organoids are grown? And I can talk about that too, but...
Go ahead.
You want me to? Okay, I will.
That was going to be my next question.
So what I like about the way Dusty described this is that it actually takes the important components of a tumor. So there's not just tumor cells in there, there's supporting cells. And there's immune cells. You've probably heard of immunotherapy. Those actually grow in these organoids, but on the 2D plastic dishes, they're not going to grow. So this enables us to more accurately mimic what's happening in the patient. So you can think of it as like this microcosm of the tumor that we can actually grow in the lab and study and control and understand. So there have been extensive studies outside of even our labs in Europe, across the US, everywhere, where people have shown that things like genetic expression in the original tumor is recapitulated in the organoids. Drug response is recapitulated. People have shown a few prospective studies, lots of retrospective studies, that these organoids really tell us what's going to happen in the patient. Whereas previously, people were taking tumors from a patient and trying to engraft them into a mouse, and those were very unsuccessful, also very costly and very long-term, so it would take 6 months to get a readout. So now you can see how it's really important that we can grow these things in the lab, and then the next big question is how can we use them to create better options for patients?
How long does it take to get an organoid from the biopsy, having an organoid that you can actually work with?
It depends on the cancer.
Yeah, so it's highly variable. In general, we're very good at most of our cultures growing within a week or two. But a lot of that depends on what we've received, and what the patient has received. So if a patient has undergone radiation or chemotherapy prior to us receiving the sample to be grown, that makes it a little bit harder. Interestingly, primary tumors, so where the cancer starts from, seem to grow better than tumors that have metastasized or gone other places. A big reason for that is the actual amount of cancer cells. So in the primary tumor, there tends to be a lot more cancer cells than stromal cells, whereas when the cancer has spread, there tends to be more kind of supportive or scar tissue-like cells in the tumors themselves, which makes it a little bit harder to grow, but we can grow them. So anywhere from 24 hours to, for some tumor types like neuroendocrine tumors, which is a very slow-growing version of cancer in many cases, those actually can take months to fully establish.
What about the brain? Can you make organoids from cancer?
Absolutely, we've done that. So both from cancers that started in the brain and cancers that metastasize to the brain. So we have now been able to grow over 15 different cancer types.
So do you folks do a DNA analysis of every organoid that you establish?
So we don't do it on every sample, because we have done enough testing now where we know what is in the patient's sample is the same as what's in our organoids. So many times the way this works is if people are going to get genetic testing done and get a biopsy of their tumor to have that genetic testing done, the same time they get the biopsy for the genetic testing, we get an additional biopsy for the organoid culture. And so sometimes we use that clinical genetic testing as characterizing our organoids. But what we're really excited about now is actually looking at how the cancers evolve related to treatments. So we can now treat a patient's cancer in our organoid culture, treat it with the same therapies that they're getting in clinic, and see how that changes their cancer over time. And what we're now doing is validating that how the cancer evolves in the cultures is what happens in the patients themselves.
Yeah, I think this is actually really a great example of personalized medicine. That's what this is called today, right? Because we know that the patient's tumor changes over time in response to treatment, but the problem is predicting when we need to augment or change their treatment and what the new treatment should be. So we know it's going to happen, but the problem is predicting when it would happen and what to do when it happens, right? And I wanted to actually make one little distinction about genetic testing and organoid testing, which we can talk about too. We talk about this a lot. So genetic testing is great because there are some gene modifications that can be targeted with specific treatments, and usually those treatments work really well. But then the question is if there's a genetic change that we can't target with a drug, or if the tumor is responding differently and we don't know why based on genetic testing. So I think there's two components. When people think about personalized medicine, they usually think about genetic testing, which is excellent and a great way to start. But I think the next wave of testing that we'll do will be beyond genetic testing, right?
So what we're talking about is very similar to what goes on now with infections. So if you get a bad pneumonia or a skin infection, you go in, you see your doctor, they start out with some antibiotic that they think is going to help, but then they also do a culture and they figure out which bacteria is causing the problem and which antibiotics are most likely to benefit you in curing that infection. And so we're hopeful to be able to use this technology to do a very similar thing for cancer patients. So for example, if a patient comes in with pancreas cancer and we see that their particular version of this cancer has a mutation that we think we can target with certain drugs, that the organoid culture can also help us test whether or not that drug is actually going to work for that patient. And if we see that that drug's not going to be helpful, well then we could potentially screen a whole panel of drugs to figure out what is the best drug, what is most likely going to be the combination that works for that individual patient. Because when we're dealing with cancer, we're talking about a lot of side effects of the treatments and we're talking about limited time for many patients. And so making sure that we're making the best out of every situation we can for our patients is really important.
So how many organoid cultures do you do for cancer? Must depend on the size of the biopsy, first of all. One, or do you do multiple organoids?
So for most patients, we get a limited amount of tissue such that we can only do one culture per patient. For patients who undergo a surgery, we get larger samples and we can grow multiple samples from individual patients.
Find analysis that there is a single DNA sequence for any heterogeneity in the tumor?
Heterogeneity is my favorite word. Yeah, that's why I exist. So we do find, and we see even in imaging of people, with the tumors in their bodies still, that there's a lot of heterogeneity in a tumor. So especially in pancreatic cancer, there's a spatial heterogeneity. And someone described it to me once, there's a lot of those supporting cells and then the cancer cells sort of in islands throughout. So it can be difficult to treat because it's difficult to give drugs that penetrate all of those supporting cells and then get to the cancer itself. But within the organoid cultures, it's actually really interesting because most of our studies so far indicate that we can preserve the heterogeneity of the original tumor even with a little biopsy and outgrowing these cancer cells in 3D cultures. So that's great for us because that means we can start to ask questions about how the heterogeneity of the tumor is going to affect its treatment response. So that means that a subset of the cells are likely to respond to some of the drugs, and another subset are likely to respond to other drugs. So that's where this fractionated treatment is really useful, which Dusty described. It's similar to how we treat infection, where you would change the treatment regimen after you develop a resistant set of cells. So this is becoming, I think, another really important component of personalized cancer care, and where we're thinking more about heterogeneity not only between patients, but within a patient, and how we can use that information to treat them better.
When we're talking about heterogeneity can mean a lot of things. So it can be heterogeneity mean differences between patients. It can mean differences within the same cancer. And when we're talking about differences, it could be differences between cell types, differences in genetics of the cancer cells. But also what I think is kind of underappreciated and where Melissa's technology really helps us is understanding the heterogeneity in the metabolism, or how active the cancer cells are. Many of our typical chemotherapies that we use are really dependent on how metabolically active the cancers are. And a lot of the techniques that we used to use to assess that treatment response didn't really take into account that metabolic heterogeneity, whereas our organoid cultures, and especially Melissa's technology, can really help us with that.
Yeah, I guess you're pointing at me, that means I should talk about
Yes.
that. So what we've done, it's basically a microscope, so if you come visit my lab sometime, you'll see this microscope and it takes up, an entire room, but essentially it's a microscope. So all it's doing is using infrared light to image the cells. And what we're lucky in that there are these molecules in our bodies and they naturally fluoresce, and they are heavily involved in creating energy and building blocks for new cells. So these are NADH and FAD. They're metabolic cofactors. So if you remember back from your chemistry class, maybe in high school, you might remember oxidation reduction reactions. So basically it's all about donating an electron to an enzymatic reaction or accepting an electron for an enzymatic reaction. So it happens that these ubiquitous metabolites are present in the cells and they naturally give off light that tells us about what they're doing. So they're telling us how many of these molecules there are, but also what they're doing. So their decay time, their fluorescence lifetime, it's a photophysical event. Basically, it tells us if the molecule is folding and not taking part in an enzymatic reaction, or if it's extending and taking part in an enzymatic reaction. So that's really specific information that we get, and it's great because it tells us about how the cell is reacting to the drug by how it's changing its reactivity and its metabolic pathways. So we do that, and then we don't have to actually perturb the cells at all, so we just look at them with infrared light, and then we get these images, and we can segment single cells, and then we can ask, how is the distribution changing? So you might have one subpopulation of cells that responds to the treatment and one that doesn't, and then you watch how that changes over prolonged treatment. And the nice thing about these organoid cultures is that we can do this relatively inexpensively, so I think the next challenge really is to automate things like growing the cultures, which takes, a research tech, as you mentioned, with some pipettes and a hood, and that is great for research purposes, but long term, it would be good to automate that. And then you need things that can automatically image those cultures and tell you quickly how the patient might respond to different potential treatments. Yeah.
Change in the DNA ever culturing then?
Yeah, so Dusty's done lots of these experiments where he'll look at the evolution of the,
So where we see changes, and this is very similar to what happens clinically for patients, where we see changes in the DNA of the cancer cells is when we specifically target alterations in the cells. So if someone's cancer has a mutation, let's just say in a protein called epidermal growth factor receptor, so EGFR, we have drugs that target mutations in EGFR, and patients respond really well when they have a mutation in EGFR and we use one of these drugs. Pretty common for us to do this in lung cancer, per se. What we find is that when that cancer becomes resistant to that drug, it changes its DNA. So it can change in the EGFR protein itself can change so that the drug doesn't bind like it used to, or sometimes we find completely new mutations in different parts of the cancer cell actually come up because of these treatments. And we can see that in our cultures as well. So when we treat with what we call targeted therapies, or therapies that are designed to go after specific mutant or changed proteins, those therapies over time will induce other mutations to happen, or at least select for cells that have other mutations.
I will say something completely random, and because I was just thinking about the evolution of the cultures, and something Dusty and I have been thinking about is taking advantage of the multiple cell types present in the organoid cultures. So it turns out that interactions between not only tumor cells, but immune cells and supporting cells are what ultimately determine the response of the tumor. So now that we have this system where we can keep all of those cells alive and together and interacting as they would in a tumor, we're starting to ask questions about what novel therapies can be used that can take advantage of these interactions. So this has been a really fruitful, I think, collaboration. It's still really early in its stages, but we're not the only ones thinking about this, but people are starting to understand that we can use these organoids to not only inform treatment decisions, in the clinic now, but we can use them to try to understand new weaknesses in the tumor and perhaps new classes of drugs or immunotherapies that could be used on different cancer types. So even with the non-standard therapies, there's questions about what is the best patient population, what features do the patient have to be eligible to use different treatments. And I think the organoid cultures can help us start to understand what patients would match with what experimental therapies as well, or therapies in clinical trials that you're thinking about.
So an example of that is we've been interested in what chemotherapy we could combine with some of these new exciting immune therapies. So people often see commercials for Keytruda or Opdivo, these new therapies that try to harness the body's own immune system to help fight the cancers. And there's been a lot of research done of can we take those immune therapies and combine them with chemotherapy, and what's the right chemotherapy? And thousands and thousands of patients have gone on clinical trials with these immune therapies and these chemotherapies. And what we're able to do with our organoids now is look at when we treat our organoid cultures that have different, so fibroblasts, which are some of the supportive cells in the culture, and other immune cells like macrophages and T cells and neutrophils, all these things in the culture, what we find is that we can actually predict what is going to happen to these other immune cells when we use the chemotherapy in our dish. And so an example of this is a chemotherapy for a particular GI cancer has been used to try to stimulate immune therapies for patients. But when we put it in the dish, what we find is that it changes all the immune cells and the fibroblasts to actually a more resistant phenotype. So it's basically protecting the cancer cells from the immune therapy. And so by having better models that allow us to better understand the tumor biology, that's going to lead us to have better clinical trial ideas. So we know that we shouldn't use that chemotherapy with the immune therapies because it does have some benefits on the cancer cells, but there's a lot of other cells in the tumor microenvironment or around the cancer cells that it's having negative effects on.
Yeah, and in the same vein, I think we're learning a lot about basic metabolism of these cells. So, if we add a therapy to, say, a culture full of fibroblasts and some tumor cells, we'll see the fibroblasts responding in really interesting ways. And those are non-malignant cells. So then the question becomes, how are these non-malignant cells supporting the tumor cells so that they are resistant to the therapy? And so the same question, that we're working with him on are the distribution of the metabolism, not only of the tumor cells, but of the immune cells, of the supportive cells, the fibroblasts. And a lot of these immune cells, as you were saying, can change to be more immunosuppressive. So that's what we're trying to avoid when we're giving immunotherapy. We want to have a hot tumor, we call it. So we want to have tumor cells that are recruiting immune cells so that there can be a more effective attack in that way. But if you're trying to study this in a human, it's really difficult to look at single cell behavior, single cell recruitment, immune cell behavior. And so the organoid cultures allow us to ask these questions that we just can't ask otherwise in a mouse or even in a human. So it's really helpful.
How many cells do you have in an organoid?
Well, let's see if we could do the math. It depends on the culture, but the organoids we look at with Dusty, they're probably 200, 300 microns in diameter, so a few hundred cells. But they can grow big if you let them. So we tend to keep them, we'll split them and keep them at smaller size. But that's actually another advantage of the organoid cultures. If you want to grow them bigger, you can start to create gradients of oxygen. So, you can picture a 1 millimeter mass of cells. It's harder for the oxygen to diffuse all the way through to the center. It's also more difficult for those drugs to diffuse all the way to the center. And it's hard for glucose to get there. It's hard for waste products to get out. So then you start to see something that looks more like a tumor, where you have these gradients of oxygen and glucose and waste products, or pH, that are building up. And then you start to ask different questions about the microenvironment and how that affects drug response. So these are all things actually that are active areas of research that people are trying to control and understand to see how we can create the most realistic model in our dish that predicts what's happening in the patient.
So, put a drug into a patient. That drug, that the drug is going to the tumor, but oftentimes the drug will be metabolized before it goes to the tumor as well, right? Prodrug.
Potentially.
Does that happen? And
So it all depends
how can you control for that in your organoids?
So I think the major difference when you're talking about metabolism of drugs in patients versus our organoids is that our organoids don't have livers. And so that is the big difference. And so what we are restricting ourselves to is using the most active metabolites of the drugs in the culture. Because if the most active metabolite of the drug in the culture at concentrations that we can achieve in patients, if that doesn't kill the cancer cell in our dish, it's not going to have any effect for the patient.
Yeah, and actually you've hit on something that people ask me all the time when I give scientific presentations. They'll say, how is it possible that you can take this tumor out of the patient, put it in this completely artificial environment of a jello mold, at body temperature with, some CO2, and then say it's really telling me anything related to what's happening in the patient? And there's several reasons. So one is that we're actually maintaining the multiple cell types of the original tumor in this little microcosm of a tumor. I like to think of it as a microcosm because it doesn't have blood vessels. As you mentioned, it doesn't have a liver. Hormones, you can control with the media components, but there's not these cyclical variations in hormones. But still, those multiple studies keep showing that these little organoids really do predict what happens in the patient. And we think, part of this is it's programmed into the cells. It's part of how it interacts with its environment. But this is also an area of investigation, so we're trying to understand it. But for the most part, things like drug delivery are tough to recapitulate in an organoid. But still, the overall drug response is very similar to what we see in patients. I don't know if you have thoughts on that, but I get this question every time I give a talk, even from scientists in my field who are doing the same research. And it's a tough nut to crack, so we haven't quite figured it out. But
So it unfortunately turns out that a lot of the chemotherapies that we use for patients are actually very modestly effective at actually killing the cancer cells. And so for many of the patients. And so what we see in the dish is very similar to what we end up seeing in the patients, even though it's in a completely artificial situation. But if you're not able to kill the cancer cells by putting, doses that we can achieve in the patients of those drugs on the actual cancer cells, it doesn't matter what the microenvironment's doing, it doesn't matter what the immune system's doing, it doesn't matter what the liver's doing, any of this. If you can't actually kill the cancer cells with the doses of the chemo or radiation that you're doing to those cultures, it's not going to have benefits for the patients. And so we've now have a cohort of patients where we have actually treated their organoids with exactly what they got in the clinic and have been able to compare responses. And so to date, we are great at predicting if a therapy is going to be resistant. So nearly 100% of our patients, if our organoids say it is going to be, the treatment's going to be resistant, the patients don't respond to that therapy. And it's about 70-80% of the time if our cultures would predict response that the patient actually has a clinical response based on their CAT scans or MRIs that they have in clinic with their oncologists.
Ever change the treatment on the response of the organoid then?
So that is our immediate goal of what we want to do. So the issue is that this has been kind of brought up through the research system, and if we are going to actually decide what to do for patients, we need to have a test that has passed this CLIA certification, basically make it a clinically usable test. And so there's a number of benchmarks that we have to jump over before we can actually change a patient's therapy based on the cultures.
So you haven't been able to do that yet, just for regular?
Correct. So we have done correlative studies where we treat exactly the same thing that the patient's getting in clinic in their organoids and shown that we can predict response, but we can't randomize a person to standard of care therapy versus what our organoids say yet. But that's where we're going. I mean, that is the immediate thing that needs to happen.
And of course, since I'm an engineer, I'm thinking about throughput issues, right? So if we want to be able to test every patient, I think it's really important to develop technologies that can automate that process. So we're working on that too.
Could we follow up on the technology, because you said that's one of the goals of getting this whole thing into.
Yeah, I have this dream of a box where you just like take the tumor, and you put it in the box and the organoids grow, and then you push another button and you assess their drug response, And actually it's not crazy, but I think it's possible because, if you think about the cell therapies people are developing, there's actually, if you've heard of CAR T cell therapies, there are these systems called the Prodigy system, So all you do is you take a leukopak from a patient and you thaw it and you put it in this box, and it adds the reagents, it adds the stimulation factors, and it allows you to test every so often, and you're essentially growing in a bioreactor these cells. So that's a different story, right? Because it's a suspension of cells, it's like blood. But they're able to automate the process, and that's reducing the cost of CAR T therapies and all those things. But I think it's possible, so I think we got to try, because I think it's
So there is hope.
Of course, we're researchers. We always think there's hope, and I think that the key part is that we found a way that's accurate at actually understanding what's happening in the patient, and that's the intellectual hurdle that I think was lacking before that we've now crossed, and so the rest of it is like an optimization problem, which is what engineers love.
So how many patients do you typically use this for in a week?
So not that many in a week, probably anywhere from 1 to 5 a week. We have cultured somewhere around 300 to 400 patients' tissues.
Certainly is going to be a method in the future for.
Absolutely.
So maybe open up the floor to questions that the audience might have. Please wait for the microphone. Question up here.
Once we had this box where we would just throw it around. It was like a box microphone, and you could chuck it across. People are scared of that.
My question has to do with accessibility. Will people be able to get this kind of personalized treatment routinely, or will it be so expensive that it will be available to the majority of people? And if so, is it really ethical to advertise treatment?
I can take that on. So so personalized medicine, we are already doing. So we are now offering free genetic testing for patients. Any patient with advanced cancer who sees a physician at the Carbone Cancer Center can get this testing done for free through our collaborations. We're also collaborating with groups across the state. So we have helped Gunderson Lutheran and La Crosse also have a similar program. We have helped the Marshfield Clinic have a similar program, and we collaborate with those doctors and other doctors across the entire state and into Illinois, Iowa, even as far as South Dakota and Georgia are collaborating with our molecular tumor board, such that we can look at the genetics of the patient's cancers and understand their cancers in a new way that help us provide treatment. So that we are doing already free of care. The organoid culture, as its current state, we're not advertising as a tool that we can use to predict response for patients, but we are really excited about its possibilities in the future, and it is currently a very valuable research tool that's helping us develop new treatment strategies, helping us identify types of patients that may benefit for particular clinical trials. But we're not to the point where we can offer this as a clinical service for patients. So we're by no means offering that, though I would love to be doing that very soon. With our current techniques, a patient sample doing a small screen of select therapies right now in our lab, because we have to have budgets for grants and things, I actually know what the cost is, and it's about $12 per patient to grow the organoids and treat them with a drug.
And I think you said once, I don't want to speak for you, but the recommendations made by your precision medicine molecular tumor board, they're usually covered by insurance or always covered by insurance, right? Your recommended treatment plan based on genetic testing.
So our recommended treatment's based on, so what we do is no matter where a patient gets this genetic testing done, their treating physician can send their report to us, and we review their report for free. And we send their treating physician a letter that outlines what we would recommend for that particular patient. The patient doesn't have to come travel us so if this is a patient up in Green Bay, they don't have to necessarily come see us to get our recommendations. We can go over their case, discuss it with a panel of experts here from pharmacy, genetics, pathology, medical oncology, et cetera, et cetera. And we actually send the recommendation back to their treating physician, all done for free. And then that letter can be used by their treating physician to help advocate for the patients actually getting that treatment through insurance. Amazing.
And it's supported by the state of Wisconsin, right?
Right.
Yeah,
So the support for this molecular tumor board is actually a budget item in the state, so they help support that.
I was wondering, how long does it take for you to be able to determine whether or not the drug had an effect on the organelle culture once you put the drug in that culture? How long?
1 or 2 days. Usually we do a 2-day readout, but sometimes you can get away with 1. So that's nice because it's a clinically relevant time frame. So you asked before how long it takes to establish the cultures, and it's very quickly, a couple weeks and then a couple days for treatment response. So that's just about in the sweet spot for actually helping people. Yeah.
So where we are right now is that we think for about half of patient samples, we would be able to give them a result in 2 weeks.
I have a question about your magic microscope.
I like that.
Is there a specific frequency of light that you find is important? And can you tell us more about what the effects are
Yeah, I can get geeky on you. So it's 2-photon microscopies. That means, usually when we excite a molecule, we use 1 photon to excite it to its excited state. But with 2-photon microscopy, we want to use less damaging wavelengths, near infrared wavelengths. So we use 750 nanometers, which allows us to excite NADH. And then we ask how long does it take for that molecule to decay back to its ground state. And that depends on who that molecule is binding to and how often it's binding to another molecule. So that again tells us about the enzyme binding activity. So this is the fluorescence lifetime of the molecule, and it's about a nanosecond. So that's 10 to the minus 9th seconds. So that means we have to use photon counting electronics. So we have to time the arrival of photons with respect to a laser pulse that's going at about 80 megahertz, so every 12.5 nanoseconds. And that type of precision is what we need to be able to make our measurements. That's why it takes up a whole half of a lab, because we have to time the arrival of photons, we have to count single photons, and then localize them to picoliters in the sample.
So there's a,
Yeah, you were talking about heterogeneity of cells. And I had a question across the original tumor to the metastases. Do you see big differences in the cell types between those? And do you grow out separate cultures for those?
I mean, yeah. So we get samples from metastatic sites and we get samples from primary sites. And they look different originally, and then they look different after treatment. So it depends on the type of treatment they're getting. You could probably say more about this, but.
So when we look at the cell types, there's a dramatic difference between the cell types that are in metastases and the cell types that are in the primary tumor. When we look at the genetics of the cancer itself, if the patient hasn't been treated, the genetics in the metastases are almost always identical to what we find in the primary tumor. Now, where we find differences is if the patient's been treated with something. So treatment has a bigger impact on the genetic heterogeneity than the metastasis process does.
but this sort of brings up another question that I was going to ask is, you obviously don't sequence the entire DNA of these cells. You must limit it to particular target genes that are most likely to be involved. Is that correct?
So we have done whole exome sequencing on our cultures, but we actually prefer to do kind of what we call DNA hotspot sequencing, where we look at those genes that we know to be most cancer relevant, because that allows us to do deeper sequencing or get a better representation at that particular spot.
So how many genes do you normally target for sequencing?
Our standard panel is about 500 genes.
500 genes.
You said you typically get one organoid per sample. How do you then do a bevy of tests or a sequence of tests?
Oh, he was mentioning he gets a dish, basically. So you get 10 to 100 organoids per biopsy.
I would say most samples we get 100 to 1,000 organoids per sample.
But it's all.
It's all contained in one culture system.
And so you could treat each one of those separately?
Well, we can also expand them. So we can let them grow for a week and then, for lack of a better way of saying it, mash them up and make them grow new ones. And so we've had some patient samples that we've been able to grow for years by just continuously letting them grow and break them down, and then let them grow up again, and then continue to break them down.
So two
He's
more
been
questions.
waiting. I feel bad. Yeah.
So, I think I heard you say that you have to get CLIA certified in your lab before any of this is, I guess, used to benefit patients. So what are like the regulatory hurdles that you have to get through before this happens, and how long do you expect that to take?
So it is a 1 to 2 year process, likely, assuming things go well. And so a lot of what we're talking about is, one, similar to what we're already doing, showing that our results, when we get a patient sample, actually predict how the patient's going to do in the clinic. But for CLIA certification, it's actually more important that let's say we get the same patient sample 3 times, that we give the same result all 3 times. So that type of validation we need to do to make sure that our assay is as robust as it can be. That way, anytime we get a similar sample, we get a similar result.
So, that's the last question here.
Dustin, you used the term solid cancer, and you gave breast and pancreas and lung as examples. Are there tumors or cancers that are not solid, and would lymphoma be one of those?
So we typically think of solid versus blood-based cancers, and lymphoma is kind of at the border between the two. So in some cases, lymphoma is more of a cancer that's free in the bloodstream. In many ways, it also can be like a solid tumor, and our Strata, our molecular tumor board, is open to patients with lymphoma as well.
Okay. Laura says we have time for another question.
I'm just wondering if you've done some research on figuring out what triggers these mutations.
Not me, no. Have you?
So this has actually been studied a lot, and there are many factors in which mutations can occur. So typically we think about viruses and carcinogens, just certain chemicals causing these things. But probably what causes the vast majority of the mutations that are present in a cancer is age, meaning that every time a cell divides, it makes at least 3 mistakes. Now, many of those mistakes are either repaired or in places that don't cause an abnormality that would lead to a cancer. But most of it is kind of the natural errors that happen with cells turning over as people age. So an example of this is if you take a patient who's in their 30s with colon cancer, their cancer probably has somewhere on the order of 40 to 60 mutations, most of them not actually leading to a growth advantage. Whereas if you take someone in their 80s or 90s that has a colon cancer, their cancer probably has somewhere between 100 to 120 different mutations that are present. Just as we all age, our cells can make mistakes and mutations occur. And it's actually amazing to me when you think about how many cells are in our body and how many cells are turning over, that we actually don't have more cancers. It's pretty amazing how well our body is able to defend off of cancer.
And just to comment on that, it almost sounds like it's a mutation, not mutation, but you make cheese, there's mutations of fermentation. Yes, we gotta kind of renew it. Because
I don't know
right
how
now
to
it's
make
like
cheese.
a rogue fermentation. Way off track.
I'm sorry, I think I only heard like every other word, so I'm not quite sure. I heard cheese and fermentation.
I know in cheese making, every once in a while you get a fermentation that just goes way off track. I just wonder if that might be a cancer. It just goes way off track.
When they're
Like a
growing
reaction.
really rapidly?
Yeah.
That sounds like a good question for you.
Yeah.
I'm not sure.
I'm a winemaker, but not a cheesemaker.
All right.
One last question. Somebody's eager. Yeah.
Oh, she's eager.
You have to wait for the microphone.
There's rules here, you know.
So drug therapy is what you use to test in the lab. Do you use radiation therapy as well? You do.
Yes. So Melissa and I published a paper together very recently demonstrating that this technique is good for chemotherapies, targeted therapies, but also for radiation. So one of the things that I was thinking was like, okay, you take this little tiny dish and you have this tiny little organoids, this little growth of cancer in this dish, and we're going to expose it to the same doses that we give patients in the clinic of radiation. It's like, it is going to wipe out every one of these. Like, how could this ever be the case? And what we saw looking at patients with colon and rectal cancer was that some of the patients' samples, the organoids responded as you would expect. They shrunk, they died. It was obvious that they were going to benefit. But we had some that, despite high doses of chemotherapy, higher than what we can give patients, in addition to giving 2.5 times the dose of radiation that we would give a patient at one time, the organoids continued to grow.
Presumably they have good repair mechanism or something.
They must, yeah.
Well, this has been extremely interesting for me, and I hope for the rest of the audience as well. And I want to thank our two panelists here.